It's a pleasure to introduce Dr. Xingjun Fan, who is working with me, and he has played a major role in the generation of several animal models of accelerated aging, and that is what is he going to talk about in reference in particular to the aging of the human lens.
It's always nice to have the annual meeting so we can discuss our research on campus. Today I can talk to one of the mouse models to recapitulate the age-related nuclear cataract. So before I get started, I want to thank people involved in this project. So the animal carried the major work task. The first three people was most of the take care of the animal, and I'd like to thank Ben Lian for his proteomics work and Kathy for the histology. I'd like to thank Michael Robinson for providing us the cream mice, and of course the leadership from Dr. Vincent Monnier. So our laboratory for many years focused on the research of post-translation modification, oxidation of crystalline in pathogenesis of age-related nuclear cataract, especially in nuclear sclerosis. Here, the lens picture, you can easily see the different opacification, which is graded by called period grade. So this lens discoloration was positively correlated with crystalline aggregation and cataract formation. So for many years researching in the field, I guess we basically accept the two major hypotheses which cause age-related nuclear cataract. One is post-translation modification. We also can call the carbonyl stress. Another can come from oxidant stress. The chronic oxidant stress can also relate to the age-related nuclear cataract formation. So our laboratory, from many years of research doing the chemistry of behind those post-translation modification, now we also start focus on oxidant stress related to this research. So we tend to create an animal model to recapitulate this cataract formation. So why we need to do this research anyway for the cataract? Because you always can say, well, you can do the cataract surgery when you have cataract. So for be a basic scientist, we want to first understand what the mechanism causes age-related nuclear cataract formation. But a second time, the long-term goal, so we want to, this is the age-related nuclear cataract formation with the age of human being. But in our long-term goal, we want to delay this onset, which if 5 to 10 years delay of the onset, I personally believe most of people in their lifetime are not necessary to do the cataract surgery. So in past couple years, Dr. Monnier presenting this retreat, so we already showed, established, create a transgenic animal model to recapitulate those carbonyl stress. One of the animal model we created is transgenic ascorbic acid transporter transgenic mice. These mice show the lens ascorbic acid level in these mice are similar to the human lens. In 12 months old, you can still see this lens discoloration with almost similar in 67 years human. So we also found those have high ascorbic acid cause the glycation modification for crystalline. Today, I'd like to report in another part of our research with oxidative stress induced age-related nuclear cataract formation. But when we talk about oxidant stress in the lens, it always means that it's an impaired system. You have a impaired antioxidant defense versus oxidant stress. They have high oxidant stress in lens. So one of the most abundant antioxidant in the human lens is the glutathione. This has been also shown that glutathione is impaired in the aging human lens. I like particularly you focus on the right side, on the red lines, you can see this. From young age to old age, the glutathione in different portions of the lens are dramatically decreased, especially the nuclear side of glutathione. You can see that 10 to 20 years younger lens have higher glutathione level, but when you're 50, 60, you have a dramatic decrease of glutathione level, up to 6–7%. I like to remember this number. So from Transgenic research, he demonstrated their lens have a barrier, so they can prevent such as glutathione penetrate into the nucleus. So that's why you can see always nuclear glutathione level is much lower than cortex. Glutathione work as a network and antioxidant network. It's working with many cofactors to detoxify free radical in vivo. And we also know that glutathione is responsible for the keeping ascorbic acid in a reduced form. From our transgenic animal, we found if ascorbic acid goes through this oxidation form, will quickly have metabolized, produce carbonyl stress. And also, glutathione is needed for detoxification of dicarbonyl compound, such as methylglyoxal. So while the methylglyoxal modified compound called methylglyoxal 100 imidazolone is highly present in aged humans, especially in cataract. So glutathione is a cofactor work with two enzymes to detoxify the methylglyoxal. So with this in mind, we created those LOX-P Cree system, conditional knockout lens fiber epithelial glutathione de novo synthesis GCLC. So from this knockout mice, apparently the GCLC, which is responsible for glutathione synthesis, the activity almost abolished in the homozygous knockout mice. This knockout mice has a reduced glutathione level from 50 to 70%, which is similar as aged human lens. And the GSH, which oxidized glutathione level, worse GSH ratio, which is the oxidant stress index, was almost double in the homozygous mice. So we also found those homozygous knockout mice, we give the name Lexco mice, have a high methionine oxidation. It's indicated the oxidative stress is increased in the Lexco mice. But we did not see any change in terms of glycation, post-translation modification glycation particularly. So when we first got this data, we got a little bit surprised, but then we realized months in the lens glucose level in the lens is much lower than serum. So plus the ascorbic acid level in mouse lens is almost nothing. But in human, you have also the low glucose level in lens, but at the same time you have high ascorbic acid level. So we anticipate when under higher oxidant stress in human lens, ascorbic acid could go to oxidation at the damaged crystalline. This is exactly on our future study. We're going to breeding these Lexco knockout mice with our previous created transgenic mice. So we can create an animal model with both oxidative stress and carbonyl stress. So next we want to check the protein disulfide formation because high oxidant stress erythropoietin can cause disulfide formation. We use this called traditional two-dimensional SDS page system. This is a typical way to do. First, you run the first dimension with no reducing SDS page. Then you cut this strip and you run a second reduced SDS page gel. If the protein form interdisulfide formation such as BC or EE, you will see in the right triangle, you will see the protein, this intermediate form interdisulfide. If the protein form intradisulfide, such as F and D, you will see the left triangle. If the protein without disulfide formation, you will be in this diagonal protein. So this is, we did those mice in 1 month and 20 months old, and the same thing the Lexco mouse, 1 month and 5 months. So first, let's focus on the interdisulfide formation. In one time, you can see the 1 month to 20 months, you can see dramatically increases the disulfide formation. Similar thing with Lexco mice. In the 5 months old, you can see the almost even higher number of disulfide formation compared to 1 time 20 months old. So for the intradisulfide formations, a very interesting result we found is a very young length. The most of disulfide are intradisulfide. When they get aged old, this intradisulfide disappeared and shifted to the interdisulfide formation, similar as our Lexco mice. So with the help of Ben Lian, we identified most of those proteins which form the disulfide formation. So such as most of the region A are gamma crystalline, region B are beta crystallines. And very interesting to see that this one line of band, most of the identical crystalline, just different oligomers. So why this always gamma crystalline and beta is more easy to have those disulfide? It totally makes sense because this, such as gamma 6 crystalline, they have 9 cysteine residue. Alpha crystalline only have 2 cysteine residue. And from those crystal structure gamma crystalline, we found that the cysteine residue, most of cysteine residue are buried. So, but when the Lexco mice, we start to show this disulfide formation in the gamma crystalline, that indicate that gamma crystalline start unfolding as denaturation happening. This may be explain the mechanism of why the nuclear cataract formation, because gamma crystalline has done unfolding and denaturation that cause aggregation. So this is from slit lamp, the 1 time, 4 months, 9 months old, you can see that kind of clear. But Lexco mice at 9 months, you can see the severe nuclear cataract formation. This is, we track those cataract formation in terms of age. So we can see the Lexco mice accelerate this opacification cataract formation compared to white type. So let me summarize this. Homologous Lexco mice, like old human mice, have a 50% drop reticular glutathione level. Lexco mice demonstrate high oxidation, such as GSSG, worse GSH ratio, and methionine oxidation. Lexco mice develop age-related nuclear cataract. I believe this animal model will be good model for the pharmacologic agent screening. Thank you very much for the presentation.
Thank you.